DNA from unusual sources helps ancient human family tree reconstruction. Neanderthals congregated in a cave 3,000 feet above present-day Spain's Atapuerca mountains 100,000 years ago. Cooking and watching children wrestle left a trail of DNA in the form of skin flakes, spittle, and other body fluids. For millennia, dirt gathered and guarded genetic material, even as people and animals sought refuge there. Since 2008, researchers have discovered a plethora of stone tools and animal bones at the Estatuas site. For DNA analysis, the only Neanderthal bone identified was a toe tip with a Swiss cheese consistency. As a result, the genetic characteristics of the residents and their connections to other Neanderthal tribes could not be determined by the researchers. Instead, they dug through the soil looking for stray DNA. Sediment from strata that developed between 70,000 and 113,000 years ago was gathered by researchers in excavations at Estatuas in 2018. Over 800 silt samples were taken from Chagyrskaya and Denisova caves in southern Siberia that contained DNA-bearing remains of Neanderthal and Denisovan ancestors in 2017 alone. Benjamin Vernet and his colleagues at the Max Planck Institute in Germany were able to isolate human DNA from that of animals, plants, and microorganisms. They found that a treasure mine of genetic information may be found in soil that is free of artifacts and fossils. Over 220 silt samples from Denisova cave yielded human DNA fragments, compared to 9 remains containing genetic material. Neanderthals, Denisovans, and Homo sapiens all resided in the area at various times, according to the soil. As with other fossil investigations, these findings confirmed the methodology suitability for Estatuas, which lacked DNA evidence from the past. New Neanderthals arrived roughly 100,000 years ago and genetically varied from the Neanderthals who had lived there for the previous millennia. And major precedent was established by these disgusting discoveries. Stone tools and other man-made artifacts are quite ubiquitous, but fossils, especially those with preserved DNA, are not. The mitochondrial DNA, mtDNA, found in sediment is simply a slice of the evolutionary tree's maternal lineage. Until now, that is, Vernet's team was able to collect nuclear DNA from certain samples and gather significantly more mtDNA than previous research had been able to collect. Researchers in Georgia and Mexico dug through cave soil in 2021 and recovered genomes spanning DNA from Ice Age humans, wolves, bison, and bears. Paleofeces were also being investigated at Harvard University and the Joslin Diabetes Center. The 1,000 to 2,000-year-old turds were discovered decades ago in rock shelters in Utah and Mexico and included digested food, including maize and grasshopper. DNA from the microorganisms that formerly made up people's microbiomes was extracted by Meredith Snow from samples of sediment at the University of Montana. They recreated 181 ancient genomes and tested them against hundreds of current stool samples from 8 nations. The findings, which were published in May's issue of Nature, suggest that antibiotics, processed foods, and increased sanitation are all contributing to a decrease in the diversity of human microbiomes today.